Hi everybody. Today we're going to be looking at a couple experiments that exemplify intermolecular forces, the way that one chemical compound interacts with another chemical compound on an atomic scale. We're going to start with paper chromatography, which is using pieces of filter paper and seeing how well things interact with the filter paper versus water that's going up it. We're going to look at miscibility, which is a fancy way of saying how well two things mix, and the intermolecular forces that give rise to whether things mix or not. This is also, we'll go into more detail in that in Chem 125, 128, because there are more features to it. But we can start with some of the basic ideas of intermolecular forces. And then we'll go to gas chromatography, which is the lab that we actually do in 124 or 127. But isn't very visually interesting. Intermolecular forces is probably one of my favorite subjects because it's so applicable to our day-to-day lives. If you ever thought, hey, how does an eraser work? We can talk about that through intermolecular forces. If you've ever thought about, hey, what makes soup creamy? What makes my refried beans a good consistency? We can actually think of that in a lot of ways in terms of intermolecular forces. What we're going to do in this experiment is we're going to use a piece of filter paper as what we call a stationary phase. So this is going to be something that sits still. And paper is made out of cellulose. So it's predominantly a compound that has no permanent dipole moment. And then we'll also use water with a little bit of salt in it as a mobile phase. Water, as you know, is something that has a very strong permanent dipole moment. And by using these two things, we'll be able to experiment with, in this case, different food dyes. We'll be able to tell whether they're interacting more with the polar molecules, the things with permanent dipole moments, or the nonpolar molecules, the things without permanent dipole moments. So we'll take a look at that, and we'll go ahead and set that up real quick. So we've got 4 dyes we're going to consider, and in a lot of ways this is similar to what we did with the absorption lab, in that we're looking at dyes, we're looking at the components that make up the dyes. We're going to have some separation occur, but this is going to be experimenting with a different property of the chemical compounds. Rather than looking at the energetic interactions with light, we're going to be looking at the molecular properties of whether it has a dipole moment or not. Here is our setup for paper chromatography. We've got our filter paper. We've got some different food dyes splotched on there. You can see the blue, red, yellow, green, and I also had a black dye. And right now they're only interacting with the stationary phase, the paper. We've got this line here to see if we can tell how far things travel. And we're going to pour water into our beaker. So this is a slightly salty solution. Goes in the beaker. Our filter paper with the different dyes we're trying to figure out, go inside. We want to make sure it's not touching anything. We want to make sure the water level is low enough that it doesn't hit the dye. And then we want to leave it alone. And what's going to happen is the water is going to naturally seep up the paper just through capillary action through the structure of the paper. And as that happens, we'll see the dyes travel with the water if they're interacting strongly with the water. The further up the paper the dye travels, the more it's interacting with the water. The less it travels up the paper, the more it's interacting with the paper. So based on this amount of motion, we can actually get information about the molecular properties. Already, you can tell the blue dye is not smearing out very much. The blue dye seems to be more non-polar, that is to say less interacting with the water, more interacting with the paper. The yellow dye is smearing out quite a bit. The green dye is also smearing out quite a bit. We'll see how much it smears. So this is just going to take some time. Eventually we get to this point where the gravitational force pushing the water down versus the capillary action up hits a balance, and we can take a look at our paper more closely. It'd be better if we let this dry out a little bit before we try to analyze it, but here we can see how the colors split up. We can see that the blue and the red had some amount of interaction with the paper and some amount of interaction with the water. So they have a combination of polar and non-polar interactions. The yellow interacted with the water a little bit more, and the green interacted with the water very strongly. Here, this black dye we had before, you can now see that it's starting to split up in terms of its color components. We have some yellow component to it. You can see a little bit of red hue and a little bit of blue in the middle. And so it looks like that black dye was made of a combination of blue, red, and yellow. The important thing here is that we can use this idea of a stationary phase, the paper, and a mobile phase, the water crawling up the paper, to separate out compounds based on their interactions with the mobile phase versus the stationary phase. That is to say, when we're looking at these interactions, we're looking at the intermolecular forces between the dye in the paper versus the dye in the water. And those differences in interactions are going to allow us to get information about the behaviors of the chemical compounds that make up each dye. For the next IMF's demonstration, we're going to consider iodine crystals. Interestingly enough, iodine, I2, makes these kind of strange looking crystals when it forms together. And we're going to look at how this interacts with water versus how this interacts with hexane. But before we look at this experimentally, let's go ahead and think about what this is going to do theoretically. So to get started, let's consider what the structure of hexane, water, and iodine look like. Do me a favor, take a moment on a piece of paper, draw out these Lewis structures. I2 for iodine, H2O for water, C6H14 for hexane. I'm just going to use the line drawing, 1, 2, 3, 4, 5, 6 for hexane. And what we want to do with each of these is we want to determine, does it have a permanent dipole moment or not? That is to say, is there an asymmetry of where the electronegativity is in each compound? And if we look at iodine, oh, that pen doesn't work. If we look at iodine, we get no dipole. If we look at water, we get a dipole. And if we look at hexane, we get no dipole. So just from this first kind of consideration of what kind of energetic interactions are we going to have, are these things going to mix or not, we can look at this and say, well, the thing with the dipole is likely going to interact with the other thing with a dipole. The thing with no dipole is likely to interact with the thing with no dipole. There's more energetic interactions to consider if we really wanted to get down to how much this happens and how it happens. We do that in Chem 128 or Chem 125. But let's just, on this first pass, in considering the intermolecular forces, I would think if we just had water and iodine, the water, because of its dipole moment, would keep interacting with the water. And if we had water and hexane, these two things wouldn't mix because the dipole moment of the water would have an energetic benefit of interacting with itself rather than being forced to interact with something else. Let's take a look at that. Let's start by just considering water and hexane. So in here we've got hexane. I have it covered because it's a pretty volatile organic compound. It has quite an aroma to it that I don't want to experience too much. And what we're going to do is we're just going to carefully transfer. We'll start with actually the water. We'll put in some water. And then we'll carefully transfer some hexane in as well. And what you'll notice is you end up with two distinct layers. Hexane layer on top and a water layer on bottom. So we can see that the intermolecular forces of hexane with hexane, or rather water with water, make it so that these two things don't mix. And if I shake it up some, I get some bubbliness, and eventually it goes back to having two separate layers. Now, we're going to go ahead and add in the iodine crystals. Because of gravity, they sink right to the bottom. And you can see the iodine crystals down at the bottom just kind of sit there and slosh around. You can already see some purple from the pouring through. Now here's what we're going to do. Right? If I shake this up a little bit, we get a little bit of purple up there from the iodine crystals dissolving in the top, and no purple in the bottom. Now I'm going to shake this up a lot. And essentially what I'm trying to do is I'm trying to make it so the iodine crystal is exposed to the hexane. And when the iodine crystal is exposed to the hexane, it dissolves up there some. There's a limit to how much will dissolve if I put in too many crystals, but the more I shake it, the more I can make this layer turn purple from the iodine in hexane, versus having this crystal down on bottom where we don't really get any purple on the bottom because the crystal doesn't dissolve in water, it dissolves in hexane. We can predict this generally from the idea of intermolecular forces, and then we can get more detailed by taking into account the energies associated with it in Chem 125, 128. So intermolecular forces can tell us what interactions are likely to happen. Another way to consider this type of interaction of intermolecular forces in a more complicated type system is to imagine what happens when we end up with a compound. You'll notice from the names methanol, ethanol, butanol, propanol, hexanol that all of these are alcohols, and the prefix tells us that the carbon chain is getting longer and longer. So we can look at a small molecule with an alcohol functional group or a large molecule with an alcohol functional group, and we can look at how a molecule with a mix of polar and nonpolar components can interact with something like water. So what we're going to do is we're going to add each of these alcohols to one of these test tubes, and then we're going to add water to see if the different alcohols will dissolve in water. Before I do this, take a moment, draw all of the chemical compounds, their Lewis structures, and make a prediction. Do you think it will dissolve in water or not? What's your justification? What intermolecular forces are you looking at? So what we're going to be looking for is kind of the same thing we saw with the iodine example, where we ended up with two layers, a water layer and a hexane layer. And the question becomes, for this test tube, which is hexanol, does adding the alcohol functional group make it so that this compound will now dissolve in water, or will it still stay separated? Right? This chemical compound definitely has a permanent dipole moment in the alcohol group at one end. But it also has a nonpolar chain in the form of the hexane leading up to that functional group. So which dominates in this case? We add in some water. Ooh, that was a lot of water. So in this situation, we can go ahead and try to disturb it more. In this situation, you'll notice we still end up with two distinct layers. So even though we added a functional group to our hexane and turned it into hexanol, it still separates out. So the interactions of the water with itself are stronger than the interactions of the water with the hexanol. So that creates this separation. We can then say, well, what happens if the chain, if that nonpolar chain gets shorter? And this is looking at propanol. So in this case, with propanol, it really looks like everything mixed together. I don't see two distinct layers. We're going to let that sit for a little bit and see if it mixes back out. What about butanol? In the case of butanol, we still see two distinct layers. If we shake it up, kind of disturb it a little bit, we can create some cloudiness. We can make it so the layers separate a little bit, but ultimately they split back up. And the reason we see that difference between propanol to butanol, and actually if you look closely you can almost see a little bit, not really, is because propanol has a shorter carbon chain than butanol. Right? Propanol's the 3-carbon chain, butanol's the 4-carbon chain. So if we're putting these instead of in the order that I did them, and if you put them in order of chain length, the two things with the longer carbon chain split their layers up still, and the thing with the shorter carbon chain ends up with no layer. We can kind of deduce what's going to happen with ethanol and methanol at this point, right? If propanol mixed with the shorter carbon chain, ethanol with only 2 carbons should mix as well. We get some cloudiness at the beginning. Doing a little bit of flicking. The ethanol mixes together. I can see a little bit of waviness to it from the mixing and the difference of refractive index between the ethanol and the water, but given a little bit of time, it mixes, and we don't end up with two distinct layers like we did when we had a longer carbon chain. Last one. Methanol. Oh, I put in a lot of water that time. Again, methanol, because the short carbon chain doesn't create enough surface area for the London dispersion force to overwhelm, the alcohol group will dominate in that case, and the water and the alcohol will mix. So here we can see how a change in chain length results in a change in surface area, giving rise to a more nonpolar compound. Even though it has a permanent dipole moment, this compound still doesn't mix with water because of the energetic interactions, considering the dipole-dipole interaction versus the dipole-London dispersion force interaction. And we can also see this type of idea in a more measurable way by looking at gas chromatography. So the next thing we're going to do is we're going to look at a gas chromatographer, which takes a little bit of setting up. And we'll look at that. Gas chromatography, it turns out, is pretty similar to paper chromatography in that inside here there is a giant coil that kind of runs around and around and around, and it's chemically treated to act like a stationary phase, to act like the piece of paper. And then we inject a sample, it pulls air into it, and the air getting blown through the coil acts as the mobile phase. There's a detector at one end that can then tell us when a chemical compound goes by and what kind of signal is generated. And so this allows us to kind of separate out and say, well, how long does it take? If I could rinse water through this continuously, how long would it take to push out the green sample versus the yellow sample versus the red sample? And we would call this, how long does it take, a retention time. So the more that the column holds onto it, the slower that the chemical compound moves through, the larger the retention time, the longer it takes for it to get out. And the more that interaction with the tube, we can tell something about the chemical compound. Now to do this, we're going to have to set up on the computer. So in order to make sure that each chemical compound gets removed from the column, we're going to be using some temperature controls, and we have to make sure we get those temperature controls set up properly, which of course you all don't have to worry about at home. 80 degrees final. So while this warms up, we'll go ahead and consider the different chemical compounds that we are going to be injecting. So here are the three chemical compounds we're going to be injecting. 3,3-dimethylbutadiene, 4-methylpentane-2-one, and hexane 2-one. So these names are sometimes daunting when you first see them, but actually they're pretty easy to break apart if we know how to read them. This one's kind of the easiest. We've got a hex and an one. That is not, I know it looks like one, but we actually call it one. That's the suffix for ketone. So this is going to be hexes, like the hexane we saw earlier, 6 carbons. So we can go 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And on the second carbon, we put our ketone. Double bonded oxygen. We could have put it on the third carbon or the fourth carbon or the fifth carbon, but this tells us specifically put it on the second carbon. So that's our hexane 2-one. Breaking apart this name, on the fourth carbon, we have a methyl group, and then we have our pentane chain, that's going to be 5 long, and then on the second carbon, we've got an one, a ketone again. So the main body of it is going to be 5 carbons long, 1, 2, 3, 4, 5. On the second carbon, we put a ketone, and on the fourth carbon, we put a methyl group. You'll notice that these two structures actually have a lot of similarities. They have the same number of carbons, the same number of hydrogens, the same number of oxygens. It's just this one has more branching. So the amount of surface area is going to be reduced. Now go ahead and try to draw this last one on your own. So you start with the 4 carbon chain, and then add di is 2 methyls, 3, 3. So we're going to start with a 1, 2, 3, 4 carbon chain. Second carbon gets our ketone. And then on the third carbon, I'm going to put a methyl group, 1, 2, 3. And on the third carbon, I'm going to put another methyl group. Again, these two structures, in fact, all three of these structures, have the same molecular formula, but different orientations of the atoms. That means they're all isomers of each other. They also have the same functional group, so they have a lot of similarities. They all have the ketone functional group. The only difference between them is the surface area of the non-polar component. And so this should give rise to some difference in interaction between the mobile phase, the air that we're blowing through the gas chromatographer, and the stationary phase, the chemical treatment inside the tube. And what we're going to do, rather than having some kind of piece of paper, we're going to get a chromatograph with peaks that show us where this happens. So this is going to be a little bit tricky to do on my own, but I'm going to do my best. We actually need to use a micro syringe in order to inject the sample into the gas chromatographer, the GC. And because it's a time-based measurement, I want to make sure I hit collect at the same time I inject. Usually there's somebody here to help me with that. Okay. Gently glide the needle in. Okay, now the timing. And now we wait. We have to wait. So air is blowing through this. It's making it so that the chemical compound, the 3,3-dimethylbutanone, is being blown through the chemical tube. Depending on how much it sticks to the tube, it'll take longer for us to detect a signal. If it moves very quickly, we'll detect our signal sooner. If it moves very slowly, we'll detect our signal later. And from that rate of motion, we'll be able to determine whether the chemical compound is more polar or more nonpolar. More importantly, we'll be able to tell whether the chemical compound has a strong dipole moment interaction or a weaker dipole moment interaction. Or maybe the best way to think about it is to think about it in terms of the intermolecular forces of the compound with itself affecting it when it boils, and then the intermolecular forces of the compound interacting with the tube. And does it interact well with the tube, or does it interact poorly with the tube? If it interacts well with the tube, it will move slower. If it interacts poorly with the tube, it'll move faster. It'll interact with itself more and just stay kind of bundled up in the middle. And there we have our peak. So this peak shows us how long it took for the chemical compound 3,3-dimethylbutanone to make it through the tube. Without some additional information about what the tube inside of here is, we can't really say much, but we can use this to measure relative to the other compounds we inject. So I'll go ahead and stop that. After we've got our peak, we can stop. It's strange to me that it didn't line up perfectly. And then we can measure our retention time for this peak. And that is going to be 3.5 minutes. So 3.5 minutes in order to get that chemical compound, 3,3-dimethylbutanone, to exit the tube. That is to say, the retention time is 3.5 minutes. Okay. So for our next chemical compound we're going to inject, it will be the 4-methylpentanone. So this compound has a longer carbon chain, which in turn gives it more surface area than our first compound. That larger surface area, since they have the same functional group, that larger surface area is going to be the important difference between them. And we want to see how that larger surface area impacts the interaction with the stationary phase inside of our gas chromatographer. Now we'll just wait for this sample to collect. While this is collecting, I'll speed it up so you don't have to wait the full time for it to show up, but make some prediction based on the molecular formula, not the molecular formula, based on the molecular structure. Where do you think this peak will be relative to the first one? Will it be before? Will it be after? Is it interacting with the tube more, or is it interacting with the tube less? Oh, here's our peak. Much larger peak, even though we injected about the same amount. But if we wanted to, we could use the size of the peak and the area under the peak to figure out the amount of material, but we'd need some sort of standardization to know how each material responds to the detector. What's important for us in this is the fact that our retention time for the second peak is later than the first peak. It's down at 4.37 minutes, so it's like at 4 1/3 minutes. That increase in time means that the second compound was interacting with the tube inside of the gas chromatographer more than it was interacting than the first compound. So we can use this difference in interaction to say that this second compound has some difference in response, some difference in the structure. We can use this to then say that the tube inside here must be nonpolar. Now, how can I figure that out? Well, these two compounds, the 3,3-dimethylbutanone and the 4-methylpentanone, they have the same polar functional group. They both have the same ketone. The only difference between these two is the surface area of the nonpolar side. And as we know from the other videos we've watched this week, a larger surface area means a stronger London dispersion force. So the London dispersion force from that larger surface area is dominating in this case. We've got one more sample to inject as soon as this is ready. Hopefully you're able to predict what's going to happen with this last compound by looking at the Lewis structures associated with hexane 2-one versus the other two compounds. And there's our third peak. Each of these peaks can now be related to the amount of interactions the chemical compound has with the particular tube inside of this chromatographer. This comparison for these three chemical compounds is relatively straightforward because they have the same molecular weights and the same functional group, with only a difference in the amount of branching, which gives rise to a different amount of surface area or a difference in London dispersion force. As we consider more complicated mixtures of compounds, we'll see how a change in the molecular weight or the functional group can also influence this idea of retention time or the idea of boiling point.